Good evening, sir. Am I audible, sir? My light is not there, sir. Shall we start, sir? It's 8 p.m. now. Yes, sir. So, very good evening to you all. Shri Kumarswami Mahavidyalaya Ausa, District Latur, organized World Record Series 2. Today there is 231st day of the series, and the resource person for today's session is on dais Honorable Dr. G. Sangeetha Vani Madam. So first of all, I welcome to you, Madam, for the dais, Principal M. Betkar, sir. I welcome the colleagues from my college and all those dear participants. The topic for today's presentation is antimicrobial resistance. Now this topic is going to present as a she is an associate professor serving at Ethiraj College for Women at Chennai. So with this brief introduction, now I request you, Madam, please.
Any age, anywhere, and in any individual. So misuse of antibiotics, and that leads to the problem because so many newer infections like pneumonia, tuberculosis, salmonellosis are hard to treat due to the inefficacy of the antibiotics. And this antibiotic resistance may see this patients die difficult the US due to multi-drug resistant TB strains worldwide. And in our case, in India, one child dies every 9 minutes due to antibiotic resistant bacteria. So the deaths, you can see that when we are comparing with other major causes of death like cancer, cholera, diabetes, and so on, AMR is going to reach a higher heights. You can see AMR in 2050, it will reach about 10 million. So 10 million people are 10 million people are going to die due to antimicrobial resistance. So what is these antibiotic resistance? Why we have to take about this particular topic, why we have to think about this? Because it's a major subset of AMR. And as I told you, the misuse of antibiotics. So resistance in the bacteria takes place either by the mutation or spontaneously it can occur due to random mutations, and it can also to the organism, because the pathogens adapt themselves so that it can survive in the present scenario. So how it can adapt themselves? Either by acquiring the genetic material which will produce resistance, or altering the gene expression, or by mutational adaptations, it is becoming survival. So what is this drug-resistant bacteria? What mechanism it has? Like when we are infection, we are given antibiotics. So these antibiotics are used to treat that particular infection, which means it helps to kill these organisms. So we have sensitive strain. So these sensitive strain, when you are given antibiotics, it will kill all the sensitive strains. But there are some resistant strains which are ineffective for that particular antibiotic. So what happens? All the organisms will be, when the antibiotic is added, all the organisms are killed, but not the resistant strain. So this resistant strain start growing. So So it starts growing and multiply. And not only that, it can also transmit the genes from resistant to sensitive strain, where this sensitive strain will also get resistant. This is how the resistance takes place in the antibiotics. So we have antibiotic targets. Antibiotic targets in the sense you can see the antibiotics can target the cell wall of the bacteria, folate synthesis, cell membrane synthesis, protein synthesis, DNA or RNA synthesis. The same way, the organism also has the mechanism to resist these targets. They have an efflux target, target modification, immunity and bypass, and also by inactivating the enzyme, they can resist the antibiotics. You can see them in detail. That is, the germs, they can develop a process in which it can avoid the target, or it will produce an enzyme which can break the antibiotic, that is the drug, the target. Or they will also develop a mechanism in which it will not allow the drug to go inside the cell. It will not allow them to move inside the cell. And you can also see they have an efflux mechanism by which the drugs can be pushed out of the cell. Or they can modify the target such a way it cannot go and attach to that. The lock and key theory will not be possible. So all these mechanisms are done by the organism to resist the target, that is to resist the antibiotics. So antibiotic resistant bacteria, there are n number of organisms which can resist the antibiotics, and the commonly formed are the penicillin resistant Staphylococcus aureus, vancomycin resistant Enterococcus, multi-drug resistant Mycobacterium tuberculosis, and life-threatening last resort drugs such as carbapenem resistant Enterobacterium infections, the drugs that are treated for the life-threatening infections. So the discovery and deployment of antibiotics. So the discovery of the antibiotics started its journey from 1910 and it moves on to 1920, 30 and it goes on and on. The same way, the emergence of resistance are also happening. So the emergence is also going on, but there is a void of antibiotics. That though the antibiotics are discovered starting from 1910 and the emergence of resistance are also taking place, but from 1980, you can see the discovery of antibiotics are going down. So this is known as discovery void, and it is why it's due to the lack of financial incentive for the pharmaceutical industry so that they cannot invest any antibiotics. So this is the problem which added to the resistance of the antibiotics. So the drug resistance is a growing problem. Yes, in 2020 in the newspaper, it was written like 30 tertiary care hospitals, they have taken the samples from 30 tertiary care hospitals and which they have seen 5 common microbes that are E. coli, Klebsiella pneumonia, Pseudomonas, Acinetobacter baumannii, and Staph aureus. These are the common microbes that are found in the culture. And ICMR have written 3 groups of pathogens. One is the resistant to last resort antibiotics such as carbapenem, multi-drug resistant bacteria which is acquired in hospital-acquired infections, and drug-resistant bacteria for a small number of groups. And as I told you, why we have to worry about this? Because the colistin, which is a last resort drug where the resistance are growing, and carbapenem, which is widely used in ICUs, are also becoming resistant. So these are the problems that we don't have any drugs to treat the life-threatening infections nowadays. So in the year 2021, they are telling that it is 10% rise in drug resistance every year. So the same way, the common organism which is found in the cultures are E. coli, Klebsiella pneumonia, and Acinetobacter baumannii, and the same drugs are becoming the problem. And when we are talking about AMR, particularly in India, it is considered to be the AMR capital of the world because we don't have any medicines to support the life-threatening infections due to the resistance of the drugs. More than 7 lakh people die due to antimicrobial resistance every year. So that is why we have to have a great concern about this AMR. One will die for every 3 seconds. And the two most affected regions are Africa and Asia. So what is the causes of this antibiotic resistance? So we know how it is happening and why it is coming. What is the causes? Over prescribing of antibiotics. Even for a headache, we are taking antibiotic. Patients not finishing their treatment. How it spreads to the community. When the antibiotics are given to the animals, they develop a resistant bacteria in their gut. And when the man eat the meat from the animals, and when it is not cooked properly, then it will transmit to the gut of the human beings. And from there, it will spread to the other people also. And this contaminated animal feces will enter into the food crops, and that will remain on the food crops. Again, when it is taken by the human beings, the consumption, it is going to, they're going to maintain the bacteria in their gut, resistant bacteria in their gut. And when they are not feeling well, when they are staying at the hospital, and from the hospital, it will transmit due to unhygienic practices to the other people nearby. And also to the and also by the healthcare facility, that is by the nurse and the doctors, to the other people through their unhygienic practices. This is how it spreads to the community. And as I told you, that is from the community, like antibiotics use and other factors from the community, from the hospitals, from the agricultural usage, or from the livestock, it will enter into the environment, where in the environment, in the soils, in the water, in the wastewater treatment plant, it enters into the particular place, that is into that particular environment. So when it is entering into that environment, how it is going to transfer? So it will transfer the resistant genes either from the person to person, or from the person to an animal, or from an animal to animal. If this is the case, it is predictable. We know what type of genes can be transferred, and it can be reduced to an extent. But when all these enter into the environment, and if a new genes are going to transform, then it is really a problem. It's a more challenging to work onto it, and we don't know what is happening and how to reduce it. So this is the problem when the genes are transferred to the environment microbiota, where a new resistant factors will form. When the AR in the livestock, so how an antibiotic resistance in the livestock? Because it is going to, as I told you earlier, India is always being designated here in the case of livestock also, it is an epicenter for the global drug resistance crisis. And the main reason is misuse of antibiotics in the livestock sector also. So why and how? The use of antibiotics as growth promoters in the animal feed. So continuously they are exposing to the gut microbiota. And then poultry is high in demand. Okay, this is highly. So to fulfill their demands for both as egg as well as meat, farmers are forced to use the antibiotics as a growth promoters, and also to protect them from bacterial and viral infections. This leads to antimicrobial resistance in the livestock. Additional factors that contribute to resistance, apart from the one which we discussed: unskilled practitioners and usage of suboptimal dosage. Nearby shop, if you have any problem, we'll just go to the nearby medical shop, and we'll take some medicines just for 2 days, or two times. That's it. We'll just stop it. So these suboptimal days will also lead to antimicrobial resistance. Poor quality drugs, particularly in developing countries. If the drug is expired, we will not throw. What they will do is they will give us a donation, or they will remove that expired label, and then they'll be using it. So all these things that happens contributes to the resistance, and particularly increase in the international travel. I think this we have faced in 2019 due to COVID, where from the China, that is COVID-19, which originated in China, transferred, and it is to the entire world. So the increase in international travel carry resistant microbes from one country to another. A typical example is the resistant strains of Neisseria gonorrhea, which originated in the Philippines and Asia, but now you can see that it is spreading throughout the world. So what are the methods to detect these antimicrobial susceptibility? We have phenotypic methods, molecular based methods, mass spectrometry. So phenotypic methods to know how far it is acting against that particular organism, how far the drug efficacy is there. Molecular based methods to know their resistance, that is the gene detection, whether a single gene is involved or multiple genes involved and what mechanism is there. All these can be detected with the help of molecular based methods and mass spectrometry. So phenotypic is a functional antimicrobial susceptibility test. So with the help of this phenotypic test, we can identify how the organism respond to this particular type of antibiotics. What mg they'll respond to this antibiotic. So for that, they'll use broth microdilution, disc diffusion, phenotypic microfluidics, and digital microscopy. This digital microscopy is very much fast, where you can very much effective because the time constraint. It is less than 7 hours, you can identify what strain it is. So this is very much effective compared to that of a classical type. And genotype identification. Genotype identification means, is there any resistant gene or any mechanism for the resistance to take place? Here, how far they are acting against the antibiotics. Here, what mechanism is involved? That is the genotypic identification. For that, we have an enzymatic method, which means any enzyme is involved in it or any nucleic acid that is present, which means any resistance gene is present, one or more. All these can be identified with the genotypic method. So with the help of that, we can create a resistance profile, which is also known as resistome, where you can take this clinical specimen and the genetic analysis done, where with the help of microarray, whether a single gene is detected or they are multi-drug resistant strains. We have to check multi-gene is detected. And then the whole genome is analyzed, and with the help of next generation sequencing, we can form a resistance genes database. So with the help of that, we can find what are all the genes that is resistant to what are all antibiotics. So how to encounter? So we have studied what mechanism is it, how it is spreading to the community, to the environment, to the livestock, and how we can encounter this, how we can solve this problem. A complete understanding of the mechanism, so which their antibiotics are resistant, so that it is easy and helpful for us to design a new antibiotic to counteract the resistant threat. So here, the Tata Institute for Genetics and Society Director Rakesh Mishra said, effective and constant surveillance can help find out which antibiotics will not work and where to reduce the unnecessary use. So this is very much important, effective and constant surveillance. So by 2050, one person could die every 3 seconds if AMR is not tackled now. So what can be done? We have to tackle the antimicrobial resistance. For that, 10 valid fronts is there. The first one is public awareness. So public should know when to use, how to use, and what is the importance of these antibiotics. And also the antibiotic usage in the agriculture and as well as in the environment. Their surveillance, continuous surveillance is very much important, so that we know how far it is effective to treat that particular infection. Human capital is definitely needed to for this particular crisis. As we all know about it, they can develop new antibiotics, and fund, particularly the Global Innovation Fund is required. Sanitation and hygiene, of course, vaccine and alternatives to meet the strategy, rapid diagnostics, and the drugs should be treated or given for the necessary treatment, not overuse or misuse of antibiotics. And international coalition is required for effective action. So in the case of livestock, how we can tackle? First, we have to create the awareness among the farmers. An unregulated sale of antibiotics should be restricted. And we have to completely ban the antibiotics which are life-threatening, that is, which are used for life-threatening infections in the case of human beings. So complete ban is required, particularly in that antibiotics. And the disease diagnostic facilities should be given at the nominal cost to the farmers, and also the recommendation of the drugs of choice. Training and awareness programs so that the farmers can be educated with the good managemental practices. And we have to give the guidelines for the proper disposal of waste, and also strict regulation should be needed or provided to avoid the drainage of untreated wastes into rivers and water bodies. And last but not the least, continuous monitoring of drug-resistant bacterial pathogens, particularly the common organisms which we found in the culture, as I told you in the newspaper 2020 and 2021. So how we can reduce the spread of antibiotic-resistant bacteria in case of human beings? First, we have to follow the doctor's instruction. We have to complete the course. Unnecessary prescribing of antibiotics should be avoided in the case of doctor's side. And clean, high contact areas, practice of good personal hygiene. All these are mandatory. So the World Health Organization, they have a global effort to address these antimicrobial resistance. They set out 5 strategic objectives. So the first one is awareness and understanding of the antimicrobial resistance, strengthening the knowledge through surveillance and research, to reduce the incidence of infection, to optimize the usage of the antimicrobial agents, and developing the economic case for sustainable investment, like new medicines, diagnostic tools, vaccines, and other interventions. We can also use a nanotechnology with antimicrobial design. Since nanotechnology are biocompatible, biodegradable, and they have inherent antimicrobial property, so that's why it can target the multi-drug resistant bacteria very effectively. And they have different types like gold nanoparticles, colloidal gold, quantum dot, polymers, liposomes, nanocrystals, emulsions, and so on. So this nanotechnology are also used for rapid detection of antimicrobial resistance. Though classical approach is there, but the nanotechnology approach is widely used, and it is used for rapid diagnostics. We have nanosensors, nanoparticles, nanothermostics, etc. So this is one such an example where we use a nanotechnology, a liposomal drug, in which when it is combined with the nanosecond pulse electric field, it helps in porating the cell membrane. Okay, it targets the cell membrane and it porates the liposomal membrane and the cell membrane and helps in entering the drug into the cell, into the porated cell. So nanoparticles, they are also derived from the plants. And why they use this plants, why they have to choose the plant? Because the nanoparticle synthesis, if it is extracellular, to obtain a supernatant cell-free extract is tedious. So the use of plant-derived nanoparticles, the challenge quite easy, that is help them to overcome the challenge quite easily. And these plants are convenient, they are eco-friendly nanofactories, these plants. So to name a few, we have Curcuma longa, Catranthus roseus, Zemace, Philanthus reticulatus, and so on. But the problem over here, when we are using the plants, which is that is the nanoparticles derived from the plants, it's a problem is that their toxicity. So this toxicity limits their wide application. And current research is going on how to counteract this toxicity, which is obtained from the plants. The strategies to extend the antibiotic efficacy are to reduce the need of antibiotics, to improve the hospital infection control, to reduce the antibiotic usage in agriculture, and to avoid overuse or misuse, educate and inform the health professionals, policymakers, and the public. And we also ensure political commitment. All this helps in extending the antibiotic efficacy. So stewardship, in which case, planning and management. The planning and management in municipals, healthcare, food and agriculture, and in pharmaceutical manufacturing helps in waste management and procedures, and also into standard operating procedures, so that we can limit the use and reduce the discharges and increase and optimize the wastewater treatment. So, four lead organizations that are working on the system, multilateral system, like on human, animal, plant, and environmental health. They forced to advance. Okay, these four organizations, they joined to form a one health response on AMR. So those four organizations are UNEP, FAO, WHO, WOAH. They joined to form this one health response on AMR. So the National Antimicrobial Resistance Action Plan, NAP, the countries are agreed and they are started policies relevant to it to prevent, control and monitor this AMR as of November 2022. And they are joining and monitoring the country's progress in the implementation of NAP. The global leader group, they working together to accelerate the political action on the antimicrobial resistance, and this GLG collaborates globally so that it helps to mitigate the drug resistant infections. So AMR Multi-Partner Trust Fund Finance helps this fund finances for catalytical, coordinated policy advice, technical assistance, as well as in capacity strengthening programs. So three globally agreed targets helps in protecting us protecting us from the resistance as well as curbing the development worldwide and also helps in reducing environmental pollution. So this One Health approach, what it does is it recognizes the human, animals, environment, plants, and they are all interconnected and they are indivisible at global, regional, as well as local levels from all the sectors, stakeholders, as well as institutions. So they are grouping together, collaborating together to form a capacity building as One Health, and healthy ecosystem, healthy animals, and healthy human beings. So there is no time to wait. We have to secure us our future from the global threat, a silent threat of antimicrobial resistance. So this One Health response will protect humans, food and feed, plants and crops, environment, as well as terrestrial and aquatic animals. So we have so many recommendations in which sustainable developmental goals to curb this resistance. So fight antibiotic resistance, it's in our hands. So handle antibiotics with care. Thank you. Any questions? Thank you, sir. Sorry, sir. I think you are mute. Unmute. Yes, sir.
Yes, sir. Hello, good evening, Dr. Bedkar sir, Principal sir, Prashant sir, and ma'am. Thank you very much. Very vital subject you have selected today,
Thank you, sir.
because nowadays we are using antibiotics in ample of forms, and we are, the story has started from penicillin, and
Yeah.
that has gone to this level. Now, generation after generations, we are changing, and higher and broad spectrum antibiotics we are taking, And many of the times we are, the in Indian population, I had seen that people are going to the medical store directly, and medical store person also takes pride to
Yes.
dispense medicine without much hesitation. That you take this three tablets and three pills and three capsules, and you will
That's
be okay.
it.
But we are not thinking of long-term goal. We are developing resistance, and abruptly people are stopping antibiotics. They're taking only 1 day course or 2 day course. As soon as they start feeling better, they stop abruptly, and we are developing resistance also. That is also another very important story. So this battle between the bacteria and man will not end. It is an endless battle, what I feel. But yes, there are stories that where you'll find the person is suffering with certain infections and the no antibiotics are working. That is how what we are feeling now. So we had to go back again, to the simple and natural forms.
Yes.
That is how we can preserve ourselves and our ecosystem, and the bacterial war will get finished. So I think Louis Pasteur, he demonstrated the presence of a microbe in the air, and that has initiated the stories. Before that, we were not knowing there are disease agents. Egyptian used to say there are minute particles that lead to this condition, but now I think yes, definitely we had to see this all these aspects very important. There are some what we can say, the bacteria are also having some kind of intelligence what they are carrying with them, and it is a battle, what Darwin said, that it is a battle of life. So they are also a fight for their survival. So I think a wonderful topic you have selected, and everyone should get sensitized that use of antibiotic now and then has to be stopped somewhere. And nowadays, the things are not happening, so we are going to the steroids for again, prednisolone and vicilone, and so many steroids are introduced, and people are also without much thinking, without much logic, they are taking this kind of pills. So one has to be very careful, so that is how I think we can conquer this battle between bacteria and human beings. Thank you.
Thank you, sir.
Yes, sir. Thank you very much. And I would like to congratulate ma'am for participation in this
Thank
series.
you.
Ma'am, my question is that nowadays the construction is being changed and we are having sun blocked apartments and floors and buildings complexes. So I think that the people living at ground floors, especially, or one sided buildings, are facing less sunlight, and there are high chances of microbes getting multiplied, right? So in one study, I study about that sun reflectors are being planted in Dubai as they are constructing high level buildings, but it is not very much in common in India also. Can you please suggest some plants that produce good bacteria or good microbes, as as we all know that sun is a natural disinfectant and it kills microbes, so people living at upper floors are very active with that and they receive sunlight, but people living at ground floors are receiving very passive sunlight or in very lesser amount. So what to do apart from cleanliness, what to do with to fight with these microbes present inside the environment or the clumsy rooms, we can say. Sometimes we feel like we just open the door and okay, it's very much clumsy inside. So inside plants that can grow, can you please name them?
Yeah, you can use the plants which give oxygen, not the bacteria. Actually, good bacteria is present in your body itself. We have to maintain that. That is what I would like to tell you. Because no, the gut microbiota, the normal microflora which is present in our body should not be disturbed. We are using antibiotics. That is how it gets disturbed actually. So due to not come, I told you so many reasons. That's the thing. And in case you want, if you want to have a sufficient oxygen, you can use money plant, and you can have so many oxygen giving plants. So that plants can be used, indoor plants, which gives oxygen for you. So that is very much effective. And as usual, you have to expose yourself to the sunlight daily. That is very much needed. And there is no other go if at all we are in the ground floors, because me too in ground floor, and I know what is the problem that you are facing.
Yeah. Sometimes I feel like that plenty of sunlight should be there.
Yeah.
And as you are saying that uh, indoor about these kind of indoor plants, so the matter of
Density also reduces. That is also one of the reason. When we are coming down to the earth, there is a kind of great means accumulation of these bacterial organisms and other things. That is one thing. One more thing I want to add, madam, with your kind permission, is use of probiotics is very essential. We are having a natural probiotics with us, curd and buttermilk. So one should improve this. So naturally, our intestinal flora will have more work, and the ability which is lost because of the antibiotics, sometimes it may be recovered by this. Instead of those taking, there are some medications where we are getting those bacteria, but rather than that, I will prefer that, we should go with curd and
Yeah,
buttermilk,
natural.
and they will help us a lot. Thank you.
Yes. Apart from Bifilac, as you said, we can use a daily, I mean, natural products. That is very much essential, so that it will protect your natural microflora of the gut.
thank you, ma'am.
Sure, sir. Good. Thank you, sir. Thank you for being a part of this world record series. Shall I leave, sir? If no queries. Okay. Thank you, sir. Thank you so much.